So welcome to the second part of this afternoon's scientific program. The next lecture we're going to call the Big Easy, and I wanted to find a local scientist who could speak to our group. I called several of our members in the New Orleans area and said, you know, who would be someone great to come and talk with us, and everyone said the same thing. So I'm going to have Moises Arriaga come up and introduce our speaker, since he knows her and has worked with her very well, and it's going to be very exciting to hear her talk.
Good afternoon. It's really a privilege to be able to introduce Dr. Jennifer Lentz, who's an extraordinary scientist and collaborator, but to me really an exceptional example of taking a concept and a discovery from the bench to the bedside. I've had the honor of working with Dr. Lentz for over 20 years now, which is really shocking as I think about it. But Dr. Lentz is a professor of Otolaryngology and Biocommunications, adjunct in the departments of Ophthalmology and Genetics, and a member of the Neuroscience Center of Excellence at LSU Health Science Center in New Orleans. The overall goals of her research are to understand the mechanisms that lead to the multisensory loss associated with Usher syndrome and to develop therapeutic approaches to prevent or cure the deafness, imbalance, and blindness associated with the disease. Dr. Lentz has developed a comprehensive approach that includes a multidisciplinary team of basic science and clinical investigators to develop antisense and gene replacement therapeutic approaches for USH1C. Currently, her lab targets mutations in the USH1C gene with antisense gene replacement, CRISPR-Cas9-based gene editing therapies using patient cell lines and a knock-in mouse model of Acadian Usher syndrome type 1C. Dr. Lentz also conducts several natural history studies with Usher syndrome patients to better understand the molecular epidemiology and natural clinical history of hearing balance and visual losses in these patients and to prepare for a future clinical trial. Lastly, she hosts a biennial community symposium in Lafayette, Louisiana, that brings together clinicians, researchers, patients, families, and community members affected by this rare disease to improve clinical and community knowledge awareness of Usher syndrome. That last aspect can't be overemphasized. So here's a PhD who pretty much knows every patient's family with USH1C in South Louisiana. And this is a big deal because we have a huge incidence of deaf blindness in Louisiana, and this is the cause for the majority of those patients. So that's been really an exceptional thing to watch and to be able to participate in. So it's really a distinct honor to call her a colleague and a friend, Dr. Lentz.
Self-oriented here. Thanks, Moy, for such a lovely introduction. And thank you both, Moy and Bill, for the invitation to come and talk to you today. The Big Easy. Okay, this will practically be like a walk in Audubon Park, I suppose, to talk about what we've been doing in Usher syndrome. So, green button to advance, to forward, and is there a pointer associated with it, or? The yellow. Oh, yeah, okay. And this is okay. Oh, gosh, just touch it. Okay, so just to get us all on the same page, Usher syndrome, this is the focus of my research here. It's an autosomal recessive genetic disease that causes concurrent hearing, balance, and vision loss. So you need two mutations. You need to get one from mom and one from dad. That's the recessive part. And it affects males and females equally. That's the autosomal part. The prevalence in the world has been estimated at about 1 in 20 to 25,000 of all types of Usher syndrome. So this is considered a rare disease, which here in the US is defined as less than 200,000 people, or in Europe as less than 2,000 people. So in the world, we're talking about 400,000 individuals. In the US, about 40,000, but here in Louisiana, we have about 400 individuals with Usher syndrome. and that's because of 2 founder mutations that we know of. I'll talk about that a little bit more later on. Diagnosis still is generally made based on clinical symptoms. So the severity of these symptoms as well as the age of onset. Today, genetic testing is used to confirm the diagnosis and really determine the type and subtype. And we would say there's 3 main clinical types and 10 main subtypes. We use the numbers 1, 2, and 3 really to describe those clinical symptoms, and the letters to denote which gene a patient has a mutation or the genetic etiology. We focus on USH1C, again because of the founder mutation in the Acadian population. So this is a busy slide. It's a map of the USH1C gene with all of the mutations to date, although I should make an exception. One was reported a few months ago, so I've got to add it to the map. All of the mutations known to cause USH1C, or concurrent hearing, balance, and vision loss. I do not have the mutations on this map that cause hearing loss alone, but if they were there, they would be here in this region of the gene. The mutation we focus on is this 216G to A. It's a founder mutation in the Acadian population, a single base pair change from a G to an A, and that causes an error in the processing of the RNA transcript when this gene is transcribed. That essentially results in no protein, no functional or full-length protein expressed in the eyes or ears. So in Louisiana, USH1C is a typical type 1 Usher syndrome with congenital severe to profound sensory neural hearing impairment, early onset vestibular areflexia, and adolescent onset of retinitis pigmentosa. The Acadian type is caused by this splicing mutation, and if you look at the gene expression in our Acadian patients, we discovered that they make a truncated RNA transcript and also no full-length functional RNA transcript, and therefore no functional full-length protein. So this became our therapeutic target. Can we replace the full-length messenger RNA to get full-length protein? So we devised, the red? Yep. So we devised 2 drug development strategies using an antisense oligonucleotide or a gene replacement strategy. So when you have a gene that's transcribed, the first thing that is made is an RNA transcript. And the antisense molecule essentially acts like a molecular Band-Aid to bind that mutation and prevent the splicing defects and the translation of a faulty protein or no protein. Alternatively, we use a virus to just supply the full-length messenger RNA. The antisense preventing the splicing error, it turns out, essentially forces correct processing, the generation of full-length mRNA, and the translation of normal proteins that result in hearing balance and vision. So the first thing we did in order to develop these strategies was to create an animal model. I cloned that region of the gene with the 216A mutation, as well as surrounding genetic material, and knocked it into a mouse. And the mice also have hearing loss, as you can see by the significantly reduced ABRs and significantly elevated hearing thresholds, which are depicted here in red. They have a circling behavior, which is indicative of a vestibular loss, as well as reduced electroretinograms at all ages and late onset retinal degeneration. So we use this mouse model and have now developed these drug development strategies for Acadian USH1C. So for those of you in drug development, you'll know this is a terribly reduced or incomplete drug development plan, but it's sort of at the 20,000-foot view to give you an idea of what we're working on. It includes a research discovery and clinical drug development phase that we then would move towards filing for an investigational new drug and a clinical trial with patients. So in the research discovery phase, I would say that we have completed the target identification, the drug target identification for both the antisense and the AAV, as well as validated it in our animal model. And we're currently in this optimization phase to identify a lead clinical drug candidate and have just engaged regulatory consultants to propose any remaining IND-enabling safety and efficacy studies. We're also conducting natural history studies with USH1C patients in Louisiana and around the world, both to identify potential trial participants and define clinical outcomes that could guide a clinical trial. So to deliver both of these drugs to the ear for the treatment of deafness and imbalance, we use a systemic delivery of the antisense molecule, either an IP or subcutaneous injection, or a local delivery of the antisense or the AAV through a number of different strategies, including transtympanic or topical application of the antisense, or round window injection, or injection through the canal or the vestibular organ. In patients, we would anticipate that both of these drugs would be delivered locally. In the mice, we can still deliver them systemically, and they will reach the ear as long as they're delivered within the first week of life. So this here is to just demonstrate that our target is hair cells to restore hearing and balance, and that both the antisense and gene replacement therapy reach hair cells either systemically for the antisense or locally, as well as locally with a gene replacement therapy. So we can get our drugs to the cells of interest. Also, both the antisense and the gene replacement therapy improves both USH1C gene expression as well as expression of the hormone and protein in hair cells. So you can see USH1C mice treated with an antisense have more correct full-length messenger RNA as well as more hormone and proteins in the ear, which also we can see localized to the tips of the stereocilia bundles compared to no treatment at all. And the gene therapy here, I just wanted to demonstrate that we can improve structure of these stereocilia bundles. So a wild type set of bundles, hair cell bundles, compared to a mutant mouse with no treatment and a mutant mouse with AAV treatment. Both treatments, the ASO and the AAV, also restore or rescue hearing thresholds in the USH1C mice. The antisense therapy delivered systemically in the first week of life or locally at postnatal day 1 significantly lower hearing thresholds. After postnatal day 7, the therapy is ineffective. And the gene therapy was done through a collaboration with a friend, Gwen Jeliak at Harvard, and she delivered several different isoforms of the USH1C gene to the ear locally, and we can see that The long isoform of USH1C is required to restore hearing, and the shorter isoforms do not restore hearing. Both the antisense and the gene therapy rescue balance in the USH1C mice. We measure this with an open field analysis of circling behavior. So you can see a map of the foot pattern. Here, the Usher mice treated with a placebo have lots of circling behavior, quantitated here, the number of rotations in 2 minutes, compared to a mutant mouse treated with our antisense lead compound. We see rescue of this circling behavior if it's given systemically before postnatal day 16. Similarly, the gene therapy also restores circling behavior, but has a little bit longer window of opportunity before postnatal day 20. We've also tested VSEPs and rotational VORs after treatment with the antisense. These experiments are currently ongoing with the gene replacement therapy. So we can see Usher mice treated with a placebo or not treated at all have no VSEPs or rotational VOR gains, but treatment with the antisense here significantly improves vestibular sensory evoked potentials and also rotational VOR gains and improves phase for the mice. Okay, so in summary, delivery of either of these therapeutic approaches to the ear targets hair cells, increases full-length USH1C RNA transcripts and protein levels, improves stereocilia bundle structure, prevents hair cell death. I didn't have time to show you that. And a single treatment of the antisense transiently improves hearing and balance, whereas the gene replacement improves hearing and balance for at least 1 year, the latest time point at which we measure them in mice. So now I'm going to switch gears to just briefly talk about that we're also putting these drugs in the eyes of our mice to improve vision. Here, our target are photoreceptors in the retina, and we deliver the ASO by intravitreal injection, and the ASO gets to all cells and regions of the retina. But we deliver the AAV virus by subretinal injection, and that's because to get to photoreceptors, the AAV can't get back to the back part of the retina. So here I just wanted to say also the ASO and the AAV target photoreceptors. The ASO here is labeled in green, so we're getting to all layers and regions of the retina, and the AAV also spreads locally throughout the retina. Similarly, both therapeutic approaches improve the expression of the correct USH1C mRNA and improve protein levels in the photoreceptors. Similarly, the AAV expresses a full-length USH1C mRNA, and we can still measure that truncated messenger RNA. The ASOs improve visual function and measured by ERGs, which you can see representative ERGs after a single treatment, and then the mice followed for 3, 6, and 12 months of age. After about a decade of optimizing the ASO targeting, the sequence, the chemistry, the delivery, we're currently looking at 3 top clinical ASO drug candidates and have identified that a formulation of these ASOs extends the duration of the single treatment for at least 1 year, as opposed to the ASOs by themselves last for only about 3 months. Here we retreated, and we can keep that visual benefit going as long as retreatment is ongoing. Similarly, the AAV therapy also improves visual function by ERG. As you can see here, this first down wave is the response of the photoreceptor, and this second upward wave is the rest of the neural retina. So a single treatment at postnatal day 21, and then ERGs at 3 and 6 months of age, we see a significant improvement in the ERGs. And that's what's shown here in this cohort. Usher mice treated with an AAV have significant improvement of their A waves and B waves, as well as significant improvement of visual behavior. This is a new behavioral test that we're conducting, which places the mice in an operant box that's half hanging onto a research bench and half hanging off that simulates a visual cliff. And the wild-type mice prefer to stay in this safe zone where they can see the floor of the box, whereas our Usher mice have no preference. They don't see the visual cliff. And treatment with the gene replacement therapy improves their visual behavior. So in summary, in the eye, both the intravitreal injection of an antisense or a subretinal injection of a gene replacement therapy targets photoreceptors in the retina, increases expression of the full-length mRNA and protein levels. The antisense shows that a single treatment transiently improves vision, and multiple treatments extend this efficacy, and we've now identified that a single treatment of the formulation improves both ERGs and visual behavior for at least 1 year, as opposed to a gene replacement that a single treatment improves these functions for a year. So we're currently in discussions about both the benefits and the risks for both of these approaches with our extended internal and external advisory boards. The ASO is a small molecule, and that's definitely a benefit. Manufacturing is easier and considerably less expensive. It has an efficient delivery. An intravitreal injection is taken up by nearly all cells in the retina. We can also deliver it systemically and have it get to the ear, or locally to get to all cells in the ear. Antisense has been shown to be very safe. Treatment also can be stopped, which is something that could be important in a clinical trial scenario where unknown adverse events appear. And the antisense also leaves endogenous regulatory expression mechanisms in place, which may be important for USH1C or other Usher genes that have multiple isoforms that are expressed both temporally and in different tissues over time. So the ASO leaves all of this timing of the gene and protein expression, the isoforms, the levels, and the cell type endogenously regulated. The risks for the antisense in the eye, we've not seen these put in the ear yet clinically. Some patients develop cataracts, but this is believed to be very treatable. Another risk is that they do require multiple injections for the life of the patient, which may create an ongoing access barrier. And the other main risk is that it's mutation specific. So this is a very small target population already within a very rare disease. Meanwhile, for the AAV therapy, it's believed to be a single treatment for life. So patients like this. It is efficient delivery to a single target cell population. So for those genes that are expressing in more than one cell type, this may be a disadvantage. One major benefit is that it's mutation agnostic, so it's a larger target population that might benefit with the disease. The risks so far for an AAV in the eye, we're just learning of risks in the ear. In the eye, we have a longer history of AAVs. About a quarter of these patients are developing a ring of retinal atrophy, which is making their clinicians very nervous. But I would be remiss if I didn't mention that these patients would develop full retinal atrophy if they receive no treatment at all. The other risk is that there's an exogenous regulation of gene and protein expression. So will this cause toxicity if it's the wrong cell type or the wrong isoform over the long haul? And it delivers only a single isoform. So this is that map of the gene that I was talking about earlier. This is the 216A mutation. So this is patients with this mutation would benefit from the antisense therapy that we've designed, as opposed to the gene replacement would capture all USH1C patients. Okay, so lastly, I'm going to switch gears once again to talk about our natural history studies that we're conducting here in Louisiana. We have been conducting since 2014 a retrospective natural history study of Usher in Louisiana, really to understand the natural clinical history as well as the molecular epidemiology. I just enrolled 4 patients last week, so this number is a little bit outdated. They're all USH1C. We also have several prospective natural history studies, one in vision here in New Orleans, but also in Montreal. Dr. Rob Konakoop also sees Acadian USH1C patients. But because the gene replacement therapy would capture all USH1C patients, we wanted to expand outside of our local population. So we're now also seeing USH1C patients at Moorfields Eye Hospital in London. We're also conducting with MOI a prospective balance history in our USH1C patients, as well as a functioning vision and balance using a VR technology test. And I just wanted to demonstrate here that the vast majority of the patients enrolled in our natural history studies are carrying one copy at least of the Acadian mutation, so we're very biased with our patient population here. So we're looking to enroll patients outside of Louisiana. The vision loss study we do over the course of the afternoon of the first day and the morning of the second day, collecting data on all of these tests, and this is my clinical team with Maria Reynoso here at LSU, Rob Konakoop at MUHC in Montreal, and Mikhail Mikulaitis at Moorfields Eye Hospital. And the natural history of vestibular loss and imbalance, Dr. Arriaga here is the clinical PI of this study, and I'm also working with Chris Zalewski on developing our clinical SOPs, as well as an USH1C clinical database. And we are collecting all of these data on these patients over time. So the next steps for us, the ASO therapy for vision loss, we're currently consulting with our regulatory experts for an upfront gap analysis and pre-IND task list. We're in preparation for a pre-IND meeting planned for Q3 this year. And then my plan is to submit a U01. Hopefully this funding announcement is still available at the end of the year. But it really is to propose any remaining IND-enabling efficacy and safety studies as per this pre-IND meeting. We've just started a collaboration with David Gam's lab to develop and treat patient-derived retinal organoids with our top clinical drug candidates, as well as to complete the prospective natural history study. The AAV isn't quite as developed, so we're still optimizing this vector, both the capsid and the promoter, as well as demonstrating a dose response. And so we're much further along in our drug development in the ocular side, but we do also hope to move these therapies into the clinic for imbalance. We surveyed our patient population here in Louisiana, and they reported back to us, it was age dependent, our younger patients feel as though they have a mild handicap, specifically due to their imbalance, and the older the patients get, it moves to their reporting of a severe handicap because of their balance. And actually, the development of this in my lab really began after a comment made by Moy years ago when we were talking with Decibel Therapeutics about a drug for imbalance. And he said what he would love to have is a drug that he could put in the ear at the same time as a cochlear implant in these young patients to preserve their balance over time. So we're hoping to get treatments for all these patients for their hearing, for their balance, and for their vision. And we're still we're just starting our second year of our R01 to optimize both of these therapies in the ear for the vestibular end organs. And with that, it's a huge team working in the lab with me, as well as so basic scientists, MD, PhD students, residents. Caitlin Robillard was an MD, PhD student in my lab. She came in with an interest in ophthalmology, developed the gene therapy program in the eye, but left the lab with an interest in ENT, and she's currently an ENT resident here in New Orleans, still coming back to the lab to help us with the surgical techniques to get to the vestibular end organs in our mice, as well as a number of collaborators, clinical collaborators with the natural history study, as well as basic scientists in the development of the drugs. So with that, thank you. Thank you for your attention. I'd be happy to take any questions. How do we do on timing?
We're good. So not only a wonderful talk, great science, but also a recruiter for otolaryngology. So thank you very much. I think we have time for one question, if there's anybody that has a quick question. Heinrich?
Hey, Jennifer, great
Hi,
talk
Heinrich,
as
thank you.
always. Do you anticipate that there's going to be differences in vectorization strategy for eye, ear, and balance organs?
Yes, we are working with a specific vector, AAV vector, a serotype 44.9 with a E531D capsid mutation that allows it to spread laterally throughout the eye. But this vector does not target hair cells in the ear. We tried it. It targets neurons in our hands, in our mice. And so far, the vector that we've worked with for the ear is the ANC80, but we're currently trying other capsids and serotypes in the ear. So yes, it would require 2 different AAVs for the ear and the eye.
Right. Well, please join me in thanking Dr. Lentz for coming over and giving us a great talk. Appreciate it. It's an exciting time to be in the hearing world.